Dear Chair, dear audience, thank you for the opportunity to talk about our research, which is the role of antibiotic resistance and microbiome diversity in different diseases. Let me summarize our first project. Antimicrobial resistance poses a clinical challenge due to the ability to cause difficult-to-treat infections. And in a non-endemic region like Hungary, limited data are available on the ways and the mechanisms of the resistance spread. In both of our first studies, we have found the resistant genes located either on chromosomal or on a plasmid, suggesting the horizontal gene transfer. And these studies show that even a single mobile genetic element can be responsible for the spread of the resistant genes. So therefore, targeted genomic surveillance is essential to detect and prevent these resistance bacteria spreads. My name is Réka Meszéna. I'm a biologist and a PhD student at the University of Pécs. We have a vision to enhance patient care with targeted therapies by better understanding the microbial insights. And therefore, our mission is to investigate multidisciplinary methods for the effect of microbial resistance and the gut microbiome on patient outcomes. So currently, we have 4 ongoing projects. The first 2 is about the multi-drug resistance bacteria spread in the clinical center of Pécs. And these studies based on experimental research. The third one is about the microbiome, how the gut microbiome varies in the different severity levels of acute pancreatitis and healthy controls with the methodology of systematic review and meta-analysis. And the fourth one is focuses on the effect of the microbiome diversity on sepsis development in acute pancreatitis. So let me introduce the first 2 projects in once. Why is the antimicrobial resistance is a global health threat? It was directly responsible for 1.27 million deaths and contributed 4.95 million deaths in 2019. These bacteria can spread between the environment and humans and also between animals and humans. And the clonal spread of these bacteria is easier to detect and control, especially if it's not a multi-drug resistance bacteria. However, the resistance bacteria can transfer their resistant genes by the horizontal gene transfer. And it can even happen in one patient or between different species. The inadequate infection control and also the inappropriate use of the antibiotics may increase the emergence of these spreads. So we aim to investigate the resistance mechanisms to carbapenem antibiotics in Enterobacterales species. Once we got the sample from the clinic, we cultured the bacteria, we identified, and also we determined the antibiotic susceptibility. We used the pulse field method for the clonality assessment, and the extracted DNA was used for the whole genome sequencing to characterize the resistant genes and plasmid and integron structures. So very briefly about the results. On this figure, each blue circle indicates one isolate, and in case of a bigger circles, it means that these isolates are 100% identical. I can say that we have identified the clonal and the plasmid spread of this type of NDM5 producer E. coli, which we found in 76 patients. And in the other experimental research, in this table, I would like to draw your attention only to the species column first. Because if we see this, these are the species what were being cultured, and basically this is what the clinicians get with the antibiotic susceptibility results. It doesn't look suspicious, right? However, when we went further, the study showed that 87% of these bacteria carried the same resistant genes, and the integron structure showed that also the surrounding of the genes was similar. So in conclusion, the VGS-based studies are essential for revealing the spread of high-risk clones and even smaller mobile genetic elements to limit the spread and improve the antimicrobial stewardship. Currently, we are at the phase of finishing the manuscript, and I hope we can submit as soon as possible. Let me move further with my third topic about the gut microbiome diversity in different severity levels of acute pancreatitis and healthy controls. We heard that the annual incidence rate of acute pancreatitis is 3 million in the world, and the mortality rate is also high. Most of the pancreatic infections are due to the gut barrier disruptions, which allows the bacteria translocations. Several different scoring systems are already known for acute pancreatitis. And also we know that the healthy pancreas has its own intrapancreatic microbiome. Also we know that bacteria can migrate to the pancreas from the small bowel. And we must acknowledge that there are many different internal and external factors which affect the microbiome. So we aim to evaluate the characteristic of gut microbiome diversity in the development and the outcome of acute pancreatitis. We came up with a clinical question: is there any difference between the gut microbiome diversity in severe, non-severe AP and healthy people? We used the PICO framework, and we hypothesized that the intestinal microbiota is different in different severity levels and the healthy people. Regarding the clinical implication, if our hypothesis is true, it will not immediately bring back any changes to the patient benefit. However, the better understanding the diversity of the microbiome can initiate new research and research activities for prevention and treatment. Here you can see our systematic search, what we have conducted in 3 different databases. And after the selection protocol, and thanks to the citation chaser, we ended up with 28 eligible full text. Currently, we are at the phase of the risk of bias assessment, and we are waiting for the figures. Let me continue with our last project, investigating the effect of microbiome diversity on sepsis development in acute pancreatitis. So sepsis causes almost 49 million cases and 11 million deaths annually. But how can sepsis develop in acute pancreatitis? When the pancreas became inflamed, it increased the release inflammatory mediators and activate the systemic inflammatory response syndrome. This systemic inflammation and the hypovolemia leads to or make the gut barrier more permeable and leads to an increased translocation of the bacteria from the intestine to the pancreas. The pancreas also may become necrotic, and once this infection is established, the dysregulated immune response may develop, and this reaction, rather than the infection itself, what defines the sepsis as a life-threatening condition and characterized by organ dysfunctions. So we aim to investigating the microbiome diversity on sepsis development in acute pancreatitis. We had the research question: can gut microbiome diversity predict the development of sepsis in patients with acute pancreatitis? We would like to use the PICO framework with an additional T, like sampling the patient every day during the hospitalization. And we hypothesized that the reduced gut microbiome diversity is associated with an increased risk of developing sepsis in patients with AP. I have to say that there is no clinical implication at the study at the moment. However, if our hypothesis is true and the patient has a reduced gut microbiome, here comes the research implication to prevent the sepsis development in AP by taking early actions to maintain the gut microbiome diversity. So we just have to start to draft the protocol. We have the inclusion and exclusion criteria. The intervention is fecal or rectal swab sampling, and the primary outcome is the association between the sepsis development and AP severity. So as a summary, we have 4 ongoing projects, and I hope that we can submit the first 2 as soon as possible. Let me finish my presentation with a quote from John Maxwell: "If we are growing, we are always going to be outside our comfort zone." Thank you for your kind attention.
in the first project, with respect to those 67 patients.
76.
76, sorry. 76 patients. How were they related to each other with respect to acquiring the bug?
Thank you for the question. It's really interesting. Unfortunately, we don't know the origin of the bacteria, so probably it comes from a community and not from the hospital. And also we saw there were some bloodstream infections, also urinary, but like 30% of the isolates came from a screening, like it was a colonization of the patient.
Interesting. In sepsis, which part of the gut plays the major role? The colon and the translocation from the colon or from the small intestine?
Based on the literature, I would say the small bowel is more important.
Yes, that's what I thought of also. And in this case, how much do you think that the fecal sample represents the flora, the diversity in the small bowel?
So this is also based on the literature that this is the most common used representative sample for the gut microbiome. I haven't read about any other non-invasive sampling.
So do you think that about the third and fourth project, so do you think that in severe acute pancreatitis comes with lower gut microbiome diversity? Yeah?
Yes, so...
This is the hypothesis. And why this is your hypothesis?
So regarding our third project, so it was a question at the beginning that the reduced gut microbiome, or the gut microbiome diversity changes, is it a cause or a consequences of the acute pancreatitis? Yeah. But based on the literature, we cannot investigate the first part to be a cause of the acute pancreatitis. We can only investigate how the gut microbiome varies after the acute pancreatitis.